Welcome to UbiCODE, the European training network to decipher the Ubiquitin code. Post-translational modifications with ubiquitin or ubiquitin-like proteins, called ubiquitination, controls almost every process in cells. Ubiquitin can be attached to substrate proteins as a single moiety, or in the form of polymeric chains, in which successive ubiquitin molecules are connected through specific isopeptide bonds. Reminiscent of a code, ubiquitin modifications adopt various conformations and lead to different outcomes in cells. The key scientific goal of our training program is to identify the crucial steps regulating this language and decipher the ubiquitin code to identify predictive signatures of diseases such as cancer. This is, to our knowledge, the first consortium joining efforts to crack this code. Teams are exploring how ubiquitin chains are formed, edited, and recognized to potentially be used as biomarkers and drug targets. UbiCODE researchers are also developing many tools, some of them with a great potential for clinical applications. For example, molecular traps capture distinct ubiquitin chains that can be used to predict the response to chemotherapy in different cancer types, but also to identify drug targets or therapeutic molecules to implement new clinical treatments. A better understanding of the ubiquitin code allows to develop innovative therapeutic approaches when the conservative approach using chemical inhibitors is not working. One of the ideas is called targeted protein degradation. It involves outsmarting the cell and forcing it to destroy the pathogenic protein by using its own biological process called ubiquitin proteasome system. For a protein to be destroyed in that way, it must first be labeled with ubiquitin by a ubiquitin ligase. Targeted degradation of an undesirable protein can thus be achieved using a molecular glue that combines the given protein with the ligase. The TOMI group recently identified a CR8 compound acting as a molecular glue that forces ubiquitin labeling and therefore the destruction of the protein responsible for the pathology. As ubiquitin-mediated regulation is implicated in a wide range of pathologies, the knowledge and technology developed as part of this training network will be of use to a large scientific community. In addition to this incredible scientific potential, UbiCODE aims to train a new generation of top-class researchers and consolidate European research capacities by enhancing synergies between different laboratories and scientific disciplines. Although most of our projects consist of basic research, some of them are more disease-oriented and have a great potential for developing drugs or diagnosis tools. Our collaboration with the private sector and clinicians will be key to translating our knowledge into clinical applications through, for example, access to samples and biological models. Further support and advancement in this area will also be required to ensure continuing progress in this direction.